Welcome back for more physiology. We're continuing our story of action potentials and what they do, and we're bringing that up to muscle contractions. In particular, how action potentials trigger muscle contractions. Seems fair enough. The point where muscle and nerve meet has several names. One of them is the myo-neurojunction, one of them is called a neuromyojunction, or it's called a motor end plate. It's not that difficult. We can find them all throughout your body. And if you ever have had to deal with a muscle stimulator, we could actually put one on and we could actually trigger muscle contractions. So it's not like these are big mysterious things. When we look at a neuromuscular junction or a motor end plate, whatever you want to call it, what we'll notice is it's not going to be one-to-one, meaning it's not one neuron in and one muscle cell on the other side. In reality, you're going to have several hundred neurons and one muscle. The question then becomes, well, what are you going to do? So the odds are all of those neurons aren't going to be saying the exact same thing. They're not all going to say contract or depolarize, and they're not all going to say, inhibit depolarization. Instead, you're going to get a mixture of the two. And what your body turns out to have are two distinct mechanisms to figure out who wins. One of them is what we call temporal summation. Temporal, making reference to tempo, or the beat, or the frequency, or the rhythm. That one there turns out to be whichever signal is the most often, so it has the highest frequency, that's the winner. It has nothing to do with total volume. It has to do with how often it shows up. The other option is spatial summation, and that has to do with, well, how often do you actually dump the signal? Not how often, but how much signal gets dumped. This is a game of numbers. It's voting. It's not how often you vote, it's just how many people actually voted. Both mechanisms work in your body. It's just a matter of where you look will determine which one is used when. If I go back and examine the neuromuscular junction, if I'm trying to make the motor neuron tell the muscle to contract, we know the neurotransmitter that will be released from the axonal terminal in the presynaptic membrane when calcium ions enter through a calcium voltage gate. That particular neurotransmitter is called acetylcholine. Some call it acetylcholine. I call it acetylcholine. It's the way I was trained. And what that will do is it will bind to a receptor, which will open up sodium gates and trigger a depolarization. It's a very common neurotransmitter, and it is always excitatory. It's not one where it changes its mind depending on its receptor. Once we have bound to the muscle cell, we're going to trigger a depolarization. And it turns out on a muscle cell, a depolarization is just like a neuron depolarization. A depolarization, sodium ions flood in, go from negative to positive on the inside, relatively speaking. That'll close the sodium gates, will open up potassium gates. Potassium ions flood out, makes it repolarize, will close those gates. We're back to being negative on the inside of the cell again. That process is the exact same here. So the foldables that we made still apply to here. It's just, it doesn't look like a neuron anymore, but the process is the exact same. The catch is the depolarization in muscles can be varied. In particular, smooth muscle and cardiac muscle do not need neurons to tell them it's time to contract. They actually have leakage that occurs. So they have random leaking that allows for sodium ions to dump in, or calcium ions sometimes to dump in, and the result is we get a depolarization. Did they mean for it to happen? No, it just happened. It's random, but there's a pattern to it. We'll deal with these little guys later. So when we look at a skeletal muscle, what you'll notice rather quickly is there's a lot of stuff on the inside of them. Unlike neurons, which are basically empty bags, empty straws, muscle has a lot of stuff, has a lot of protein on the inside, to the point where we kind of shove all the other parts off to the sides. Because of this, we're going to have an issue making sure that action potential gets everywhere fast, because these cells are big. So what we have are things that we call T-tubules, which are going to be these red lines in this top picture. And what those will do is they will connect different parts of the muscle cell together. What we'll notice with that is we could have a depolarization moving right along these T tubules, T for transverse, but next to each T tubule, there'll be this whitish network on either side. And we named that whitish network the sarcoplasmic reticulum, meaning the maze inside of the muscle cell. And together we call that a triad. And these triads seem to border on things that we call sarcomeres. A sarcomere is a fancy word for muscle part. And what we notice is whenever a depolarization moves through a T tubule, the SR opens up a calcium voltage gate. Calcium will immediately flood where they are, which is at the sarcomere, and they're going to bind to a component of the sarcomere that we call the thin filament. Thin filament turns out to be made out of a whole bunch of different proteins like troponin and tropomyosin and actin. Everyone mostly talks about actin, though. It will bind, and what we get immediately is this. I'm going to narrate part of it. So if I look at the sarcomere, what I see are these parts. The big thing in the middle is what we call thick filament. The blue lines are thin filaments. The things that look like Z's are called Z lines, which are the borders to the sarcomere. And what we notice is the Z lines get pulled into the center like a massive game of tug-of-war, just it's a two-sided tug-of-war. What we get is an interaction between the thin filament and the thick filament. And they kind of look like little walking feet. I showed you a video earlier in the year where we had feet that were walking around and they were carrying a vesicle and you all giggled and laughed at it. Those are practically the same thing. So we have depolarization, which will release calcium ions from the SR, which will bind to components of the thin filament. That will allow for the thin filament, or at least the actin, to interact with the thick filament component that we call myosin. What will happen is they will touch. The moment they touch, it's called a cross bridge. The moment they touch, what we'll get is a pivot. The name of that pivot is called a power stroke. The catch is you get one for free. After that, we need energy to reset the actin and myosin cross bridge. So what we do is we add ATP. The job of the ATP will be to cause the myosin to let go, reset, and do it all again. And as long as you have ATP, we can keep the cycle going. Provided there's ATP, that cycle of contract, 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 or power stroke continues. The power stroke is automatic. You do not need to be alive in order for the power stroke to occur. As long as we add calcium, that will allow the actin and the myosin to cross-bridge. The moment they cross-bridge or touch, we get a power stroke. That process, that cycle of we add calcium, we allow for a cross-bridge to exist, which immediately triggers a power stroke. The addition of ATP energy causes the cross-bridge to reset itself so that we can cross-bridge again and have another power stroke. Add ATP to reset, cross-bridge again, power stroke. This cycle is referred to as the sliding filament theory. Filaments meaning lines. And we have sets of lines that slide past each other, which shortens the muscle. This is our understanding of how muscles contract. Heart muscle, so cardiac muscle, smooth muscle, skeletal muscle. This is how it contracts. We call it the sliding filament theory because it's rather hard to test it inside of a living organism, so in situ. So it works in models, it works where we look, but really testing it inside of us is a little difficult to find out. How do we shut this thing down? We have three options. We start with the biggest and best option, and we'll work towards the worst option. First option, let's get rid of the neurotransmitter. That seems to make sense. Second option, let's get rid of the calcium. Turns out we have insanely strong active transport pumps on the SR that can pull that calcium right back in. Third option, run out of ATP. The problem with that option is we just lock your muscles in place. We actually have a name for that. especially if you're dead. It's called rigor mortis.